as we can. Okay, great. So hello, everyone. Welcome to our All Protein Dialogues webinar. This is our third webinar of the year, our third part of the All Protein Dialogues. Today, we'll be looking at the topic of bioprocessing solutions from research to application, unlocking scale in fermentation and cultivated meat. The All Protein Dialogues webinar is part of our ongoing efforts to highlight innovation across Asia Pacific, India, China, Australia, and New Zealand, and really highlighting those who are pushing the boundaries in alternative protein R&D and commercialization. All Protein Dialogues is co-organized by GFI APAC and GFI India, and as I said, it's designed to strengthen regional collaboration and accelerate technologies that can transform our global food system. So please feel free to make sure you reach out to our speakers if you want to collaborate or network with them. And my name is Arin Naidu. I'm the Senior SciTech Ecosystem Specialist at GFI APAC. I'm very glad to be your host for this 19-minute session today, where we'll be exploring one of the most important questions in our sector. So that is, how do we scale bioprocessing to unlock commercial viability and ultimately price and scale parity for alternative protein products? And achieving scale requires innovation, not only in our biological systems, but also in bioreactor design, automation, tissue fabrication, downstream processing, and much more. So today's speakers, we have three speakers, Dr. Minghao, Dr. Lee, and Dr. Vineet. I'll introduce them before each of their talks, since there'll be quite some time for us to cover and to make sure that this is fresh in your heads. But we're really grateful to have all three distinguished speakers with us today, and really spans academic research to at universities and research institutions to industrial level bioprocessing. So we're coming in with a very wide perspective on bioprocessing solutions. And as I said, huge thank you to our speakers. Each speaker will have around 20 to 25 minutes, and we'll take questions and answers at the end. So whatever questions you have, please feel free to submit your questions at the Q&A box at the bottom of your Zoom screen. And kindly make sure to mention the name of the speaker you want to direct your question to. We'll keep an eye out in the Zoom chat. We'll bring up the questions for the last part of the talk. So yeah, please be patient and stay for that part, because that's always really exciting to get their perspective. So for today, we'll start with Dr. Minghao Nie, then followed by Dr. Shilei Lee, and conclude with Dr. Vineet Gupta as our speakers. So I'll start with a brief intro for Dr. Minghao. Dr. Minghao received his PhD in mechanoinformatics from the University of Tokyo in 2017, and is currently a senior assistant professor at the Graduate School of Information Sciences and Technology. His research focuses on microsystems, additive manufacturing, biofabrication, and biohybrid robotics. And he led the development of three-dimensional skin tissues for robots, resulting in the world's first skin-covered robot with self-repair capability. This series of work are reported in major media, such as Nature and Innovative Technologies. And he also developed microfluidic devices for vascularized tissue formation and hollow fiber bioreactors, enabling centimeter scale perfusible cultured meat, seeking to build foundations for large scale tissue biofabrication. And this leads us to the title of his talk today, which is thick cultured meat fabricated using hollow fiber bioreactors. With that, I will pass the mic to Dr. Minghao. Thank you again, and we look forward to your talk.
Yeah, thank you very much, Ari. So I will start sharing my screen. So is it okay? Everybody can see my screen? Okay, great. So I will just start talking about the topic that I wanted to cover. So the topic of my title is thick cultured meat. I think the main focus of this webinar is about protein sources. But when we talk about protein sources, when we talk about cultured meat, I think we always wanted to think about the real meat, which is not only protein, but structured protein. So we are focusing on trying to create this kind of a structured meat using technologies. And this work is done led by me, but there's also other team members. So these are the team members that helped get this work done. So to introduce this, you guys probably hear the introduction of me. You heard that I am from the information science sector, and you probably will naturally wonder why I'm here. Okay, so because we are doing information science and we are initially very much interested into robotics. That's why when we look at robotics that things can move, we are naturally interested into the various types of things that can move. And if you look at biology, you know that specific muscles can move. For example, we are particularly interested in skeletal muscles. So in definition, it is the muscles that's attached to the skeleton, of course. And what it is really interesting for us is it is a very voluntary muscle, which means that you can generate motion through a neural stimulation, which means that if you wanted to create a robot, or let's say cyborgs, you must be able to control them. So that's very interesting for us. But when we do the research, we also realized that if you look at the animal body, you will know that muscle is a very, very massive organ in animal body. So that makes it a very, very major food source. That's why we are here talking about these kind of topics. So again, if we can have those cultured muscles reconstructed, we can build cyborgs like things like Terminators or something like that. Let's just allow the science fiction to fill up our imagination. But we could also build cultured meat, right? So then let's dive into how we can build this kind of muscle structure. So to think about how to build it, we have to look at what muscle tissue really looks like. So if you have a piece of muscle here, and if you're trying to zoom up and look at the cross section of it, you will see it has a lot of bundled myofibers, okay? And if you look at this myofiber into the nanoscale, you can see these kind of like a sarcomere structure. So these structure are the real structure that enable the muscle to move. And we are also hypothesizing that these structure might lead to the structural characteristic of meat. Okay, so this is the actual stained image of a cultured muscle. I hope everybody could remember how this looks like. So these striped patterns are really the holy grail that we are trying to achieve when we are talking about making structured meat. So if you look at my slide and at the left side, we know that, so if you remember the characteristic of it, it can move, right? So which means that we need to make aligned and differentiate the cell, allow them move. And at the same time, because meat is a very massive tissue, so we have to make a very massive organ so that we have to make macro-sized tissue. So we have to reach these two requirements to do a really good job. And look at the state of the art, the tissue engineering fact, is how we are making muscle. So this is the most standard way of making muscle tissue. What you do is you use myoblasts and you prepare hydrogels. And what I'm saying is, if you look at the illustration on the middle, you see this pinky jelly stuff. If you look at inside, you will see these are the myoblasts. These are the cells that are going to differentiate into myofibers. And these cells are being loaded into hydrogel. And this hydrogel are basically jelly, so containing proteins, okay? And with this setup, we add two whitey pillars here, which we are calling them anchoring structures. So with these structure, we can build the fundamental setup for making artificial muscle. And what will happen is really interesting. If you have this setup, and if you started to culture the thing, you will see what happened. The pinky hydrogel will start to shrink. And this is a characteristic enabled by the cells trying to grabbing the protein, shrinking them. And once they are shrinking, these two whitey pillars play a very pivotal role. So they are trying to resist the shrinking along this orange direction. So if they resist it, which means that there will be a tension generated along this direction, and this tension actually is felt by the myoblasts. So they started to differentiate and they started to align with each other. So with this, we can make aligned and differentiated cells. But if you look at the two requirements that I show you, so we probably can make align the different cells as per the previous study, but can we make macro size? That's a question. And if we dive into this question, I will say this answer is no. And why is that if you look at this piece of cultured muscle here, and if you focus on the cross section of the muscle, think about, we have a very small cross section, let's say smaller than 0.5 millimeter. That'll be very fine for the muscle to survive. But if we are talking about meat, we're talking about things on the order of several millimeter to centimeters. And what will happen if you have such a large tissue is there's always going to be necrosis because the culture medium comes from surrounding the outside of the tissue. So it takes the diffusion so long time to reach the central area. That's why it's going to die. So we are thinking that maybe the next huge challenge for us to make cultured structured meat is to trying to fabricate macrosized micro tissue, macrosized skeletal tissue, how we can achieve that. And our strategy is we build a device. The device is called hollow fiber bioreactor. And we use this trying to achieve such kind of thing. And I will explain with this animation. So if you look at this 3D file, you will see this is a piece of meat that we are targeting at, we wanted to make. And our trick all started with this blue parts. And if you look at this blue parts, it has a very sophisticated structure, but I will show you what it is trying to do. So first, if we flip the device, we will see this blue part holding the two ends of the cultured meat will have whitey fibers threading through. And these fibers are called hollow fiber. And they are very, very semi-permeable. And we chose the cutoff molecular weight to be 70 kilodalton because most of the nutrients in the culture medium can pass. And we choose the diameter to be really, really thin so that they are a little bit thicker than a piece of hair. And the material we use is PESU, a material that is really cheap because they are mass produced in the water filtration system. So we can use them and we try to use them as a artificial muscle, artificial heart vessel to deliver those nutrients. And so what we are going to expect is if you look at, imagine that this is the cross-section of the cultured meat that we wanted to make. We are going to expect that, okay, so this very, very thin hollow fiber we have here, and because it's semi-permeable, so if we perfuse nutrients from inside, and it can reach outside the hollow fiber, so making the cells basically happy around the fiber so that the cell can do their own work to form structured meat, okay? And we talk also about the alignment. So we also wanted to induce the alignment by keeping the tension. And how we can keep the tension is the blue part have structures that can grab the cultured meat so that we can keep the tension. So we expect if we do it well, we can see the myotubes, they align with the hollow fiber. So that's the expectation. And so with that, I will show you some of our results. So first, we check the influence of the perfusion condition on the tissue, because at the moment, everything was still in the imagination. We have to confirm whether it works fundamentally. So what we do is we make a device that only have one fiber. And with this one fiber, we do the perfusion culture and we look around the fiber and we ask questions like, okay, so if you perfuse, will the cells be alive first? And then if they are alive, can they form the tissue morphology that we wanted to have? So that's the two key questions we wanted to ask. So here are the results. So first, about the viability. We cultured for 4 days. That's long enough for the cells to die if there is no nutrition. So if we culture for such kind of long time, then we cut the tissue in the cross section and we stain the nuclei of the cells. And if we zoom in, we can see, so here, this is the hollow fiber, and you can see the blue dots, these are cell nuclei. So we can get an impression that more dots are surrounding the hollow fiber, okay? And if we go on quantify this thing, so we make donut regions and we count the cell density in the region. And on the right side graph, you can see that, okay, so with larger region, you will see that basically the cell density drops a little bit. But with higher perfusion rate, like the purple bars, you will see that higher perfusion leads to higher cell density across all the distance up to 500 micrometer. As you remember, I was talking about if the tissue is larger than 500 micrometer, cells are going to die, right? So this thing actually worked against it pretty well. But that is only viability. So beyond living, can they do good work, the cells? So in that case, we need to culture the tissue for even longer time. So we cultured for 10 days, and then we slice the sample and we do immunostaining to check out the protein markers to see whether the cells differentiated well. And we also check these two cross sections. So first, for the vertical cross section, you can see these are the stained images. On the top are the ones that with low flow rate, which means that the nutrients are delivered a little bit under. And on the bottom, you will see these are the samples with high flow rate, which means that the nutrients are abundant. Okay, and the left column and the right column represents different marker proteins. These are the hallmark proteins for myotube differentiation. So you can see for both kind of protein, higher flow rates led to higher expression area and higher amount, total amount of expression in the tissue. So that's good. And if we look at the morphology on the longitudinal side, we stained alpha actinin, which is the marker for the sarcomere, the structure that's staining whether the muscle can move. And if we zoom in, you will see these striped patterns formed really well. And the place where my mouse pointer is currently at is the border of the cell fiber. So we can see this close proximity of pretty good myotubes formed near the hollow fiber. So that's a pretty good evidence that we have. And we also did stimulation using electrical voltages. So you can see if we stimulate it with pulses, we can see the muscle contract like this, okay? This one is only with 4 fibers. And so right now, I think everybody agree that okay, the fiber seems to be working, but can we still scale up it? Because we only tried one fiber. There's still many works to do. So we went on to trying to see, can we scale up this kind of thing? The trick is, um, actually, these hollow fibers sounds pretty cool, but they are very difficult to handle. If you look at this video I show you, this is me holding the fiber. The fiber is very, very thin. Just think about your hair. It's close to your hair and even softer. So how can you get the fibers to thread into the intricate device? It's very difficult. So for example, I showed you that I have the blue parts and the fiber threading through in a very beautiful 3D CG, but in the reality, most of the fiber won't be easily going through. So that's a huge, huge problem for us to solve. But we have a very simple but facial trick. We make these two blue parts. Sorry, it's in gray now, but we scale them up so they have 50 holes there now. And if you look at these two parts, you will realize they are similar, but they are somehow different. So let's zoom in. So once we zoom in, you will see that the tip of every nozzle here have actually different geometry. And if we put them together, you will see the left side can match and get inside the right side one so that it can enter it. So what we can do is we can then pre-assemble these two parts so they get aligned, and then we thread the fiber. So how it works is like, so this is the assembled one, and we put the fiber in. So it doesn't require any technique. So you just push the fiber in, and then you separate. So you got the fiber threaded. So this is a very important trick that we have. So we cultured the thing for 10 days and to see what happens. And this is me removing the fibers. It's a bit laborious, but we got initially 2 centimeter size of chicken meat here. And do we keep the good distribution of do we keep the promise of the fibers with 1 to 50? So we can see that if we look at the cross section, on the left side is the sample without fusion. So you can see the blue signal, which is the MF20, is the marker protein. It's very sparse, right? And if you look at the right side, this is a sample with perfusion. You can see the very enormous expression of the proteins across the area, okay? And it's even across this whole piece of 50 hollow fiber area, okay? So we also did food-related analysis. Is it meat? Is it good? So we first try to test the texture of the meat. So these A, B, the two graph actually showing that with perfusion, the pinky curve, the hardness, the stiffness of the tissue actually increased against the control sample, which is without perfusion. That's pretty cool. And we even found that if you measure the free amino acids in the tissue, you will see the perfusion sample have much higher pile-up, sum-up amount of free amino acid. So overall, the taste should be thicker in comparison to the sample without perfusion. And also what's really interesting is if you look at the fraction of the sweetness, umami, and bitterness, you will see it actually increased a little bit in sweetness. So this is an interesting evidence that cultured meat could be sweeter. Okay. And finally, 50 wasn't very satisfactory for us. So we go on to trying to make it even more large size by using robots because we are really good at robots. We are not only making cyborgs, but we are really good at making automation systems also. So I have a very, very wonderful student. He developed a very tricky system to thread the fibers. So imagine that now we are going to thread 1,000 fibers into the system, and we cannot do it with our hand because it takes hours. So he made a ejector using a vacuum generator, which is sucking the air inside. And if we put the fiber here, the fiber will be sucked inside and ejected out, and through this very small nozzle and go into the device. I will show you the video. So you can see how fast it is. So it went in pretty snappy and with good precision. So we are right now, we have to feed the fiber manually, unfortunately, but that's already a good improvement for our efficiency. So we went on to make this 1,000 fiber array. So this is the time-lapse video showing that we are making the array. We have 1,000, more than 1,000 fibers in it. And he's a one-man factory. He made everything. He did all the gluing and all the cutting. He made the device. And then we did the culture. We prepare a lot of cells and load the hydrogel inside the device. And we started to perfuse. And after perfuse, so this is the cultured tissue after, I think, 6 days. And this is the one after removal of the fiber. Okay. So eventually, where are we now? We can make up to 11 gram of chicken nugget. The size is close to a chicken nugget, so that's why we are telling all the reporters, okay, it's close to a chicken nugget. Finally, with this device, and we published it just this year, and we got very lucky that Time magazine, they recently noticed us that they chose our work as one of the best inventions of this year. So we're pretty honored by this, but we know that there are still a lot of work to do along the way. So to give you a summary, so what we show is like we show the concept that if you have a hollow fiber bioreactor, and if you make it well, and by what I mean make it well is you align the hollow fibers really good inside. If you look at other hollow fiber reactors in the industry, in the market, they cannot align them well. So their fiber is randomly bundled, so it's not uniform. That's really not good for uh, culturing this uniform meat. And so we show that this concept is very important in making a very, very nice piece of meat across a very large range. And we found that actually the perfusion worked very well. And we can show that maybe we can make meat also, but if we can make such a huge piece of it, we might eventually be able to also make cyborgs that's been activated by muscles, maybe in the near future. So that's also one of our dreams. Okay, so with that, I would like to end my talk here, and I'm happy to take your questions.
Yes, Dr. Minghao, thank you so much. Yeah, I don't know what to say. This is incredible work and incredible research. I love how you went through like showing the cell viability, morphology data, then talking about scaling up, increasing from that 3 x 3 to much more. And then also, yeah, really cool to see the pre-assembled anchor pair system, and then how you then scaled it up even further with automation and robotics. I mean, the build-up was so clear in your presentation, and really cool to see. I myself have lots of questions, but we'll save the Q&A for the end. And perhaps later, if you would like to put your contact in the chat, if people also want to ask stuff. But yeah, really incredible work. Thank you so much for sharing.
Thank you very much.
Yeah, we'll close your part of the talk if you can
Mm-hmm.
stop sharing screen.
Mm-hmm.
And then I will introduce our next speaker, who is Dr. Li Shilei, the senior engineer from the Beijing Academy of Food Sciences, China Meat Food Research Center. Dr. Li graduated from the Institute of Chemistry of the Chinese Academy of Sciences in 2017 and engaged in R&D of cultivated meat and food quality and safety in the China Meat Food Comprehensive Research Center since he started his work. In recent years, he's led and participated in 6 national and provincial level scientific research projects, published more than 10 scientific and technological papers, and applied for 17 domestic and foreign invention patents. He's participated in the formulation of 2 national industry standards and one provincial and ministerial level science and technology award. Among them, 12 invention patents and 2 utility model patents were authorized in the research of cultivated meat related processes and key technologies. So yeah, for the talk today, Dr. Lee will talk about large-scale production technology of cell cultured meat. We're really excited to have you, so I'll pass the mic now to Dr. Lee. If you can show your video and share screen and unmute yourself, please feel free to begin your presentation.
Hello, everyone. Can you hear me?
Yes, all good, all good. You can go ahead.
Okay, let's begin. Hello, everyone. Nice to meet you. My name is Shilei Li. I'm from China Meat Research Center, which is also called Beijing Academy of Food Sciences. Thank you, Good Food Institute, for invitation to attend the webinar. My topic is large-scale production technology of cell-cultivated meat, chicken meat. My presentation will be reported by the following menu. The first part is background introduction. As you know, recent years, cell cultivated meat has been developed quickly across all of the world. Notably, United States, Israel, Singapore, Australia have approved the commercial development, which is inspiring. However, there are still many technology changes related to the industrial production need to be settled, from which large-scale cell cultivation technology is beyond one of them, because the single cell is too small to wait. Generally, 1 gram meat was constructed by more than 100 mini cells. Large-scale cell cultivation is fundamental to achieving increased yields in cell cultivated meat production. Large-scale cell culture relies on seamless integration of bioreactors with other materials and equipment, necessitating high-efficient cultivated processes. Based on the above challenge, recent years we have established an engineered cultivation process for cell cultivated meat through a strategy combining process exploration, theoretical simulation, and parameter optimizations, and validation in large-scale reactors as shown in the slide. The second part is large-scale production technology development of cell-cultivated chicken meats. Firstly, we have developed efficient extraction method technology for chicken seed cell in detail by coupling physical dissociation technology with composite enzyme preparations. A myogenic cell dissociation technology system has been developed, breaking through the laboratory extraction of cell mount levels. Within 30 minutes, up to 10 million cells with high purity and high activity can be obtained from 1 gram of tissue to meet the demand of scale cell production in the following step. Secondly, we have developed one low serum medium for the above chicken seed cell cultivation. In detail, the low serum culture formula can achieve continuous cultivation of chicken myogenic cells under 2% serum conditions with good cell growth status and a 1.4 increase in proliferation rate under 3 cultivation conditions. The SMM has been successfully applied to large-scale cultivation of chicken myogenic cells in the following step. Thirdly, we have developed an edible microcarrier by using the low-temperature induced phase separation technology. It is a 3D culture microenvironment on the surface for chicken myogenic cells. Fourthly, based on the above preparation, chicken myogenic cell cultivation conditions were developed by using the spinner flask. The inoculation method was investigated. Results show that intermittent storing made a more uniform distribution of cells on microcarriers compared to no storing or continuous storing. Meanwhile, the suitable storing rates and feeding methods were studied. Results show that increasing storing speed may improve cell distribution uniformly. But this process also increases shell stress and reduces cell proliferation activity. An appropriate feeding strategy not only enhances cell proliferation efficiency, but also yields more high cell viability in the final cultivation stage. The effect of cell seeding density on cell proliferation efficiency was also investigated. A result showed that at an appropriate initial seeding density, chicken myogenic cells exhibit robust proliferation and rapid growth rates. On the other hand, the effect of microcarrier density on cell proliferation efficiency was also studied. Results show that at an appropriate microcarrier density, chicken myogenic cells exhibit robust proliferation and rapid growth rate. Finally, the effect of medium composition on cell proliferation efficiency was also investigated. The results show that excessive high serum concentrations do not significantly enhance cell viability with high cost. Analysis of metabolic status during cell proliferation shows that after cells enter the active exponential growth phase, glucose consumption increases significantly and lactic acid production rises rapidly. Fifthly, chicken myogenic cell cultivation conditions was investigated from theoretically and experimental perspective by using design experiment DOE methodology and parallel bioreactor verification method. Results show that a fitting model describing the relationship between critical parameters and cell density was obtained, and the cultivation parameters in 5 liters bioreactor was obtained. Sixthly, chicken myogenic cell cultivation conditions were tested and verified in 5 liters bioreactor. The results showed that chicken myogenic cells achieved a 27-fold proliferation during the 11-day cultivation process. The cell density reached to 2.7 million cells per millimeters. Meanwhile, biochemical analysis results showed that the primary nutrient consumed by chicken myogenic cell during rapid proliferation is glucose, with lactic acid as a metabolic bioproduct. Timely supplementation of corresponding nutrients helps sustain prolonged high-speed cell proliferation. Last but importantly, chicken myogenic cells cultivation in 50 liters and 1200 liters bioreactor was achieved. In detail, based on a cascaded amplification strategy, ensuring consistency in key parameters such as graph coefficiency, KLA, and PV ratios. Bioreactors for cell cultivated meat production were designed and constructed at 50 liters and 200 liters scales, and a large-scale production line has also been established, encompassing CIP, SIP, steam system, and a biochemical analysis system. And then the production was realized according to the step in the slide, which are cell recovery from working cell bank, first phase amplification in 5 liters reactor, second phase amplification in 50 liters bioreactor. Third phase amplification is 200 liters bioreactor. Then cell cultivated chicken meat was harvested and prepared food as shown in the next slide. Based on the calculation of the cell amount and 10% additive percentage in the food, the new production amount can reach to 120 kilograms. This is the cultivated chicken meat products after fortification, which is sells new meat chop, gumbao, sell chicken dish, and sell meat burger. In my opinion, the flavor delicious, but our regulation institute don't approve it. So currently, it can't be sold in China. The third part is large-scale production technology optimization of cell-cultivated meat. Actually, there are still many problems to be solved to increase the production efficiency in my bioreactor, especially the 50 and 200 liters bioreactor. So the optimization was made progress. Firstly, is pedal type selection and optimization. Currently, there are mainly pedals used in the reactor, including elephant air, hydrofoil, and others. In my testament, we tested the elephant air and hydrofoil layer. We found that the elephant layer with double layer, the shear force and the mass transfer efficiency was higher than elephant air with single layer, and which is higher than hydrofoil layer. But in our experiment, we found that the elephant air with double layer and the hydrofoil layer can't achieve the high cell density because of the low transfer efficiency of hydrofoil and high shear force of elephant with double air. So finally, we selected elephant air with single layer. Secondly, is the ventilation strategy optimization, the carbon dioxide accumulation. In experiment, we found that the ventilation strategy is an important factor that influence the cell attachment on the mic carrier. As you know, the adhesion cultivation was influenced by the micro environment sensitivity. So, in order to keep stable the cultivation system, we wanted to add more oxygen to the system and less air into the system. But as in the test, we found that we can add more oxygen radio, high oxygen radio in the system, but we found that the pH of the system decreased quickly. After analysis, we found that the carbon dioxide accumulation is very severely in the system, which influence the cell proliferation. The third is the harvest area inoculation. As you know, the adhesion cultivation is very difficult because it needs more procedure to for example, we need the enzyme to separate the cell from the mic carrier. But in the suspension cultivation system, the problem isn't exist. So although the difficult, we have successfully established the continuous production procedure, from seed to harvest and seed again and harvest again. The fourthly and maybe the important factor that influence the high high efficient production of the cells. It's the medium composition, optimization, and fluid exchange method. By using the biochemical analysis, we found that some amino acid or and glucose was consumed greatly in the system. Based on the analysis result, we added some glucose and glutamine to the system. Meanwhile, we have experimental the relationship between the metabolite, for example, the amino or lactic acid to the the ratio of the metabolite and the cellular activity. We verified the limitation that cell can bear. At last, by the glucose concentration was increased to 6 micro per liters, and the glutamine concentration was increased to 6.5 micromolecular per meters. And some minor modification was made based on the biochemical analysis results. Meanwhile, fluid exchange method was also optimized. Finally, the result was inspiring. The limitation, the maximum density of the cell on the microarray was from 2.5 meters per millions per millimeter to 6 million per millimeter. The fourth and the last part is I want to make a brief introduction to our organization. Our organization was built in 1963, and in 1948, Japanese government provided free assistance to establish China Military Research Center. In 2010, the three research center merged to form Beijing Academy of Food Science. So our organization has two names: Beijing Academy of Food Science and China Military Research Center. We have after the development of the several years, we have for six research and development direction. It's animal-derived foods, alternative protein, food safety testing, standardization work, plant-derived foods, brewing food, science and technology media, and industrialization work. This is our alternative protein science and technology innovation base. The base occupied an area of 5,000 square meters. The pilot lines for cell cultivation meat and microbioprotein production are capable of achieving large-scale production verification. This is the equipment that's equipped in our base. This is our group. This is our email. If you want to communicate or commercial communication, you can email to me. Thank you.
Thank you so much, Dr. Lee. That was incredible. I think what I was thinking was how expansive the work, your work is, and how much it covers. I was just writing down like from serum culture media to microcarriers to like scaling up from spinner flasks to parallel bioreactors, all the way to 200-liter bioreactors, creating 120 kilograms of cultivated meat. But then even optimizing that with the elephant ear single layer paddle system, ventilation strategy, inoculation, harvest strategy, and fluid exchange system. It was really cool to see all the way from like fundamental R&D to like bioprocess engineering, how much your work covers. So that was really exciting. Thank you so much for sharing. And I think really incredible to see how much the Beijing Academy of Food Sciences is working on alternative proteins. Yeah.
Thank you.
Thank you for your talk. So any questions, please put in the Q&A box. And now, Dr. Lee, thank you so much. That's your talk for today. And then I will now introduce Dr. Vineet. So, all right. Dr. Vineet Gupta is a CEO of Fermex Solution LLP, in a leading Indian company providing turnkey biomanufacturing and fermentation solutions for the biotech, food tech, and alternative proteins industry. With deep expertise in precision fermentation, process design, and optimization, Vineet has led innovations that make advanced bioprocessing more scalable, automated, and sustainable. Under his leadership, Fermex develops end-to-end fermenters, bioreactors, and downstream systems that empower research to industrial scale manufacturing. Vineet is passionate about enabling the good food ecosystem through engineering excellence, helping transform ideas in sustainable food and biotechnology into commercially viable realities. And today, the title of his talk will be From Lab to Launch, Building Scalable Biomanufacturing Platforms for the Alternative Protein Industry. With that, I will pass to you, Vineet. Thank you very much.
Thank you, Erin, for such a wonderful introduction. So, so I hope my screen is visible to all. Great. Good evening, everyone. So, as introduced by Erin, my topic is from lab to launch, building scalable biomanufacturing platforms for the alternative protein industry. So, I'll just quickly take you through our through Fermex. Already explained by Arun, but I'll take just a few minutes. So we founded in 2016 by three passionate engineers, and we specialize in design and manufacturing of high quality fermenters and bioreactors. Our vision is to be the leading provider of innovative and reliable fermentation systems, supporting the growth and success of our clients worldwide. Our team is 150 plus dedicated group of engineers and professionals, all committed to deliver the best solutions to our client. Our core values are innovation, quality, customer commitment, and sustainability. Our growth curve is like going also exponential. And we have certifications to supply our equipment all over the world, like ASME, U-Stamp, and BBI, CE, and others. And then our products and services includes fermenters and bioreactors from research block modules 200 ml to 1000 ml, as shown by Ms. Lee also. So you can see 200 ml block, then lab scale 2 liters to 40 liters you can see here, then pilot scale here, and industrial scale you can see here. Then we also make photobioreactors, solid state fermenters, downstream processing equipments like tangential flow filtration systems, separation purification systems. And the features of our equipment are energy efficient design, anywhere mobile monitoring, and are tested on 65 plus quality control parameters. And we have achieved the feat of remote installation of 20 KL train of animal cell culture bioreactors for cultivated meat in 2023 and 24. And you can see this right bottom picture. This is that 20 KL train having 200 liter and 2 KL and then 20 KL. So this is a great advancement for the alternative protein. Then as innovation is our core value, so we are innovating a lot. So as you can see, we have generation 1, generation 2, and generation 3 panels, then Mimics, HMI Mimics, then SCADA software Mimics, and even the fermenter itself, you can see the difference. So, and the last one is AI generated, but we aspire to make such kind of bioreactors in future because these whole organ formation and which will not be rejected by the body itself from the same cells So I think it is going to be the future, and I think Mr. Lee and Minghao are already working on this direction. So now coming to my topic, so the global shift is towards alternative protein, and global alternative protein market is projected to be $400 billion by 2040. And if you see the current and the projected this thing, so we have currently we are animal protein is dominating by 450 to 470 million tons per year, but the growth is expected to be slow by 2050, and that is the reason we are here. That is the reason we are discussing this on this, because it is expected that as we grow in population, it will be difficult to feed the population with the same feedstock or available space on Earth, and that is why we are working on the meat production through bioreactors. And similarly, plant-based meat at current levels in 2025 is 30 to 40 million tons per year. It is expected to reach around 140 to 170 million tons by 2050. And if you can see in this graph, there is no cultivated meat or precision alternative protein from precision fermentation, because it is literally not there right now in the market or anywhere. It is just in the labs. So, but we, as per the projection, it is going to be like 10 to 15 million tons per year by 2050, and by precision fermentation, we will get alternative proteins to be like 20 to 15 million tons per year. So now coming to the bioreactor design parameters. So generally, we have, like as already shown by Mr. Lee also. So we have lab scale, then pilot scale, and then industrial scale. Like he was showing, we started with the with small vials, then to lab scale fermenters, then pilot scale 50 liter, and then 200, 1000 liter industrial scale. So here, if you see, because lab scale is for initial strain screening, process development, and parameter optimization, pilot scale for validation of parameters, testing process, control strategies, and yield consistency. And industrial scale for large scale operation with real-time monitoring and process control. So this is how it is, a scale up is like. For cultivated meat bioreactors, has been told by Mr. Minhao also that yes, hollow fiber bioreactors are also taking shape, and they are there. But the key design parameters which we need to maintain in the bioreactor, like the temperature 36 to 37, pH 7.2 to 7.4, oxygen 20 to 40% is all O2 is required, and then shear sensitivity very low, because the animal cells are very sensitive, and media complexity is very high. And in precision fermentation bioreactors, we have temperature range from 20 to 37 degrees Celsius, pH 5.5 to 7.5, oxygen variable, anaerobic. It depends upon being aerobic or anaerobic fermentation. And then shear sensitivity is also moderate, media complexity is also moderate, because the range of microbes will be there to produce the protein, special protein, food proteins. So coming to the design parameters, as already shown by Mr. Lee also, that elephant ear, this segmented, we call it segmented blade impellers. So these are basically used, and as you can see in our 200 liter and 20 kL bioreactor also, we have used similar segmented blade impellers. And then the differentiation also comes from the type of sparger which is used in this. Like we uh, generally use micro spargers, not the ring spargers, and the requirement of the air or oxygen is quite low. Like microbial, we are going up to 2 ppm, 1.5 ppm, but here we require very less, only 0.2 ppm or something like that. And then the controls and other things are quite demanding because it is very, you can say, very precise fermentation. And as soon as any parameter changes, our cells begin to die. Okay. So what we have given is for suspended culturing, not for like scaffold kind of manufacturing cultivation of the cells. But I just wanted to show you how it differs, like the bioreactor for the cell cultivation, animal cell cultivation, and normal fermenter. So you can see both the on the left hand side is also a fully automatic fermenter, and that is for microbial fermentation. But as you can see on the right hand side, it is a bioreactor for animal cell culture, and it is too complex because we have to maintain each and every parameter within a very narrow limits. So that is why, and there are too many parameters to be controlled. So that is the reason that it is too costly right now. And there, again, so many parameters to be monitored also and controlled and need to be kept within the range during the operation. So this is the main thing that right now the capex for any cultivated meat production facility is very high. Then the scale up challenge. The scale up challenges we know as already been explained by Minghao also in his presentation. Oxygen transfer, maintaining adequate oxygen levels in larger volumes, heat management, as he told about, like necrosis will happen if there is no proper channel for removing the waste products or removing these things. And and the gases also, because the CO2 needs to be removed from the system. And similarly, shear stress, uh, because we have to give proper mixing also, as well as we need to remove the things also. And also the cells are quite sensitive. We cannot mix it so much that cells die of shear. So that all the parameters needs to be taken care while yeah uh, while we are designing it. And then the main thing is the media cost. Obviously, it is too complex, and it requires like uh, bovine serum, and then these are very costly media components. And so every batch is very important right now to be preserved, because if it gets contaminated or things like that. You're not only losing the media, but you are losing a lot of money also. So it needs to be the system itself needs to be very sturdy and robust. And then process control, maintaining consistency at industrial scale, like not only at the lab level, but at at a level of 20,000 liters, or if you are going for more bigger level like 1 lakh liters or 2 lakh liters, then just think about like how we are going to maintain the consistency of all the process parameters, gradients formed for the pH, for the temperature, because single sensors cannot give you the exact values. So these are all scale-up challenges. And then regulatory compliance, meeting the food safety standards, and the things has already been told by Mr. Lee also. It has not been approved till now in China, and here in India also it has not been approved. But yes, policymakers are working on it, and I believe GFI is working on it. So current challenges are like yield consistency, very high cost, process robustness. And currently what we are making is one kind of bioreactor which is fitting all kind of cell lines. But I believe that the future bioreactor will not be like this, because what we are going to make through cultivated meat is we are going to produce the food. We are not going to produce something high value thing. Okay, we are going to feed the people. Okay, we are not going to give them some food as a as a some healthcare medicine or something like that, or some MEB kind of therapy. So the thing is, currently the bioreactors which we are using are quite close to for the production of MABs, monoclonal antibodies. But for the production of food, we certainly do not require such kind of very sophisticated, very high cost bioreactors. But at the same time, currently, we also don't have information how to go about it, how to build such systems which can be economic, which have economical design also, and which can be used for resource cycling. And yes, energy requirement is there, but yes, we can always, this is a proposition that we can shift to renewable energy powered systems, and then the cell line specific bioreactors. So coming to the biomanufacturing evolution, and mainly it is concerned with how we are going to the current needs are, what are the current needs right now. So the current needs for the bioreactor is the precise monitoring of critical parameters like pH, DO, temperature, exhaust gas monitoring, turbidity monitoring, and even dissolved CO2 monitoring. So many parameters, even airflow rates, pressure, everything needs to be closely monitored and controlled in a very precise, in a very narrow range. And reproducibility of the outcomes also, like, because as we are going through the presentations of Mr. Lee and Minghao, like they were telling us, okay, we were able to make this much, we were able to make this much through one batch of 7 days, 9 days, 12 days. So these needs to be reproducible. It should not be like one time we have achieved this. It should be every time we have achieved this. And automation is the requirement because through manual intervention, we cannot control such a narrow range of process parameters. And the cost efficiency, obviously, the optimized resource utilization. I was talking to one of my friend. He's into this cultivated meat industry only, and he actually was concerned about the high media cost for this, and one of our client is actually working on uh, on medias which growth factors which are free from these growth factors from like fetal bovine serum and other things. So the friend got instantly interested in having those kind of medias which will not be derived from the animal origin to produce the animal cells. It is like we are getting the media from the fetal bovine serum, okay, and then putting it for the culturing of the cultivated cells, animal cells. It doesn't make any sense. It needs to be from such a source which doesn't require again the animal. Okay. So now coming back to the topic, so the future needs we have is specially designed bioreactors which will be tailor designed for specific cells. Like you want to produce chicken cell, okay, this is the bioreactor for chicken cells. If you want to produce some beef cell, okay, this, because the requirement of each cell is different. So if we can have specifically designed bioreactors, we can obviously reduce the things which we are putting in every standard bioreactor. So that way, I think if we can make specifically designed bioreactors for each cell line, that will reduce the cost of the bioreactor. And resource efficiency, like co-culturing and water reuse recycling systems. So that way we can reduce the resource requirement, which is very high right now also. And sustainable energy like solar, wind, tidal, geothermal powered bioreactor systems. So because what we have to meet is that we have to make this animal cell, cultured cell meat it should be cheaper than what we are eating through the animal source right now. When we are into 2040 or 50, there, when we'll be comparing the things, I believe the cultured meat should be cheaper than these traditional meat from the feedstocks. And then the design should have only essential features, like which are required for specific cell line, or where we have already optimized the things and we don't require so much of automation. As I've shown you before also, it is too complex to handle such a bioreactor, and then having that bioreactor, it is too much capital intensive, and then producing something which is like very low value. So we have to meet both the ends. If we are producing something very low value, the capex and the opex should also be very low cost. Then only we can match those price ranges. So the thing is, like, as discussed by Mr. Minghao and Mr. Lee, like, they are working on the biology part of it, and they are already doing a great work in this direction, and many others are also doing a great work in this direction. So the point is, biology invented it, and engineering, like the people who are into manufacturing of bioreactors, good quality bioreactors, are going to scale it, how it will be done in the future. So I'm just taking you through some projections into the future of alternative protein bioprocessing. And because uh, like my topic is from lab to launch, so I think till the time it will be highly um, intensive, it is very difficult to have it in the market for the people to have it. And till it is there in the market, we cannot say it is going to be there for the years. So our whole objective is to make this technology very economical so that everyone can have it. Not right now, maybe 15, 20 years from now, it will be available on everybody's plate, dinner plate or lunch plate. So now, coming to the thing, like bioreactor revolution from 2025 to 2050. So I just considered, like, if I'm in 2050 and I'm looking back from 2050, what key breakthroughs are required to make this technology scalable as well as cheap, so that we can have it on our platter, and cheaper than traditionally available meat which is there, and feed the population, obviously. So we need to have modular cell-specific bioreactors, as I already told you, interchangeable bioreactor modules tailored to specific cell lines. This can be achieved very quickly. Within 2-3 years, it is achievable, but it depends upon from the scientist who will work on this and ask for such kind of bioreactors and changes, and tailored for the tailored specific cell lines. Then in 2032. Biointegrated sensors, so living sensors embedded in bioreactor walls for real-time monitoring. So these are the requirements, so that we can reduce the cost of sensors, because currently the sensors which we are using are very costly. And then 2035 self-regulating systems like AI-driven autonomous bioreactors requiring minimal human intervention. That is going to be required in this, because with the manual intervention, human intervention, we may face a lot of issues, inconsistency in the production, errors, and when we are saying that it is going to be food for us, so we don't want any kind of contamination or anything in that food, because it is going for human consumption, and that too in very mass scale. So it needs to be driven by the automation. And then living material bioreactors. So bioreactors constructed from engineered living materials. So if there is any problem in this, that can be that can be taken care by the material itself. And quantum optimized protein folding. So quantum computing integration for perfect protein structure control. So these things will be there before we come to meat, exactly meat like thing which we are going to eat in future. So what it will do actually, it will have a good economic impact. It will reduce the cost of of the bioreactor by 80%, which is there in 2025, which we are making right now. Then 50% less water usage, 90% energy self-sufficiency will be there. So these things are going to happen when we are going for these kind of changes in the system. Then the then the engineering breakthroughs which are required, material science innovation, like from using self-healing biocompatible polymers, which can extend the bioreactor lifespan to 20 plus years. Then programmable smart materials adapt to different cell types automatically. Then living materials bioreactors, as I've already told you, grow and repair themselves. Then process control revolution, quantum sensors, which will be like 1,000 times high precision than the sensors which we are using right now. It may take 5 or more years to be available for us. Then in 2024, neural network process control. I hope everyone have heard of neural network. So they will predict and prevent issues before they occur in the bioreactor. So there'll be a series of bioreactors which will be controlled automatically through central system, and not just through a PLC or this kind of system, but is controlling a lot many equipments and taking the data from each and learning and resolving the issues before they occur. And then fully autonomous systems, zero human intervention required. So that kind of systems we are going to see, but it will take some time. Like it is, it will take like 15 to 17 years. And then energy revolution, like bioluminescent energy harvesting, engineered microorganisms generate light and heat, which is required for the bioreactor. Then thermoelectric gradient system, which will convert temperature differences to electricity. We all know when there is any bioprocess, it generates heat. And if that heat can be, currently what we are doing is we are using the water, chilled water, cool to cool it to maintain the temperature. But what if we can use that heat which is being generated to produce electricity? And then photosynthetic integration. So so algae systems can generate the energy and oxygen for our systems. So that slide is also there in the coming slides, like how algae can help. So this is there only. So co-culturing revolution, algae and mammalian cell systems. So there's a very good research by Mr. Yuji Haraguchi et al. Their team. So what is going to happen? Like why I'm describing this, because what we are making today is not necessarily going to be there for in from 10 years from now in the same field. The same bioreactors will not be used. If their work is going to be taken up, like algae mammalian cell co-culture system solved critical challenges in cultured meat production, like oxygen provision. So algae is there, which is going to provide the oxygen to the animal cells. And as per their research, it is able to make the tissue formation up to 200 to 400 micrometer thick versus normal 40 to 80 micrometer. And then nutrient recycling. So chlorella species BDH1 strains don't compete for glucose, create circular waste to nutrient system. So whatever waste is generated by the animal cell culture, it will be used by the algae, and then it will take up the waste system and produce the media again, which is required for for the production of the animal cells. Then waste reduction. Microalgae consumes 91 to 93% of ammonia, reduce lactate levels, and extend culture longevity by 3x. And then economic benefits, reduce FBS dependence. As I already told you, fetal bovine serum dependence. If it is not required, then you won't require a fetal bovine serum. First, you need to have fetal bovine serum. You will take that serum from the fetus of the bovine, and then using it for the culturing of the animal cells, maybe for beef or whatever cells. So that dependence will not be there, or it will reduce it and increase growth performance by 80% and lower production. It will, what I was talking about is reducing the cost of the media. So now why?
Dr. Vineet, sorry, I'm
Yeah,
so sorry. Would you be able to wrap up shortly? Super interesting talk. It's just we're running short on time. We've passed the 25 minutes, and we do want to get one question per speaker. So if you don't mind, if you could wrap up
yeah.
yeah, thank you.
I'll take very less time now. So just want to show this to you. So this is like taking up the cells from the muscle, and then stem cell extraction, and then serum replacement with microalgae, and then it is uh, put it put up into the bioreactor, and then cell proliferation using the bioreactor, and the waste which is generated is again recycled through the algae culture, and then the then the scaffolds co-cultivated with microalgae, and they are used for making the myotubes, which were being discussed by Mr. Minghao also. And then they are used for industrialized processing and packaging, and in for the hamburgers. And so coming back to this, so yeah, so moving on. So yeah, so we also need microbial consortia engineering. So this is apart from the bioreactor. This is like what we require from the biologist. Microbial consortia engineering is required. CRISPR enhanced host strains are required, which can produce 10x higher yields with reduced metabolic burden, metabolic pathway optimization, synthetic minimal cells, which will produce zero waste byproducts through complete pathway control. And the environmental impact will be like 75% reduction in land use, 85% reduction in greenhouse gas emission, and 90% water efficiency improvement. And similarly, the economic transformation, like by 2035 at par with conventional meat, 2040, 25% cheaper than conventional meat, and 2050, 50% cheaper than conventional meat, which we expect it to do. And cell line revolution, like general purpose, what we are using is general purpose strains. We can have specialized protein factories, synthetic minimal cells, then metabolic engineering breakthroughs we require, dynamic pathway regulation, compartmentalization of compartmentalized metabolism, self-optimizing networks, and then genetic design also, single gene editing, genome scale redesign, de novo genome synthesis. So these revolutions are required for this cultivated meat to be there, and then real time then in process efficiency, we require real-time metabolic modeling, predictive AI, process control, self-optimizing systems, and resource utilization, closed loop water systems, nutrient recycling, and energy capture systems. And in the productivity, like today it is 14 days, which can be reduced to 7 days in 2040, and then 3 days in 2050. Yield like 20 grams per liter to 400 grams in 2050, and cost reduction from 100% if taken today, then 10% in 2050. And then the cost, like it is costing approximately $50 per right now, so per kg. So to be equivalent with the currently traditional food we have, it should be somewhere around $2. And and the scale, currently the scale is like 10,000 liters. So maybe we require 1 lakh, 1 million liters by 2050, single bioreactor, 1 million liter. So a lot of challenges are there to make such a huge system and control all the things, all the parameters. And then in the infrastructure also, like currently we require a lot of space for it. The facility cost reduction will be there from 500 million to from now to 25 million in 2050 for the same facility. Vertical integration, like 70% reduction in supply chain, and digital twins, predictive maintenance and optimization, blockchain traceability, complete transparency from cell to consumer, and yes, facility like neighborhood production pods. So instead of mega factories having centralized production systems, we can have neighborhood production pods, which can give the supply, which can resolve the supply chain problems. And then the environmental revolution, obviously, resource cycling. If we are using 100 liters today, it needs to be reduced drastically up to 2 liters. Energy transformation, if we are using 50 kilowatt, it needs to be like only 1 kilowatt. Then land use, if we are using right now 10 meters square for such a system, it should be 0.1 meter square. We can use vertical bioreactor farms, underground production facilities, or space-based production. And then collaborating for bioreactor evolution. So the bioreactor design cannot be possible without the collaboration with the scientists who are actually working on the biological part of it. We can only work on their directions, like this kind of system is required. And for that, we like currently we require process engineers to optimize existing bioreactor systems, biologists to develop cell lines for general bioreactors, and policymakers create regulations for current systems. And then for future collaboration, we require bioreactor designers, create cell line specific systems, and then renewable energy experts integrate solar or other kind of renewable energy into our systems. And then sustainability specialists to design resource recycling systems. And at last, I thank GFI India, GFI APAC, GFI EC, and collaborators for giving me this opportunity to present this talk. Thank you so much. And if there is any question, I'm open to it.
Thank you so much, Vineet. It was really great to learn more about Fermex, understand the difference between the bioreactor or bioprocess parameters for cultivating meat fermentation. And I really enjoyed how future forward a lot of your talk was focusing on what are the innovations which are still required. Would you mind, just closing your screen share? And if I could get
Yes.
Dr. Minghao and Dr. Lee to open up their screens just very quickly because we are short on time.
Right.
Yeah, Dr. Vineet, you can still keep your screen on if you could just stop sharing your screen. Okay. So, I think your screen is still sharing if you can, possible for you to stop sharing the screen. Okay, great. Thank you so much. Thank you so much for all the talks from all three speakers today. Unfortunately, we are very low on time. So thank you so much, everybody, for the questions you have put in the chat box. I think Dr. Lee and Dr. Minghao have gone ahead and answered a lot of those questions already. So thank you so much to both for going in and inputting. Unfortunately, I want to be respectful of everybody's time, so I don't want to extend the call much more than this. But perhaps if we want to just get like one last thought from each of our speakers, maybe what you're most excited about moving forward, and then we can wrap up. Just like some short remarks for the audience. But again, thank you for the questions. Sorry that we're a little bit short on time. Maybe we'll start with Dr. Minghao. Yeah.
Okay, so I think Vineet talked about a lot about the processing and the future perspective about this whole industry, and I think that we are not quite there yet, and there are still a lot of open options about it. But I think eventually these things could be intertwined. Like we are thinking that maybe protein are more important, or either the structure are more important. But eventually, if you develop both, maybe we will meet somewhere in there. That's my feeling. So yeah, that's my take for today. Yeah, thank you very much.
Thank you. Thank you, Dr. Minghao, if you would like, you please feel comfortable to share your email in the chat,
Oh,
and then
sure,
our team
sure.
will
I'll do so.
pass it out. Yeah, in case
I'll do
people have interest in contacting Minghao. And then Dr. Lee, also thank you for your talk and showcasing all the large amount of work your team is doing. Any last words for the audience or anything you are excited about for the future? Would you like to share? Oh, you're muted, so if you could unmute your audio.
Sorry. Thank you. Thank you again, GFY, to provide the good opportunity to communicate with all the um, scholars or commercial attenders. I think in the future, the cell cultivated meat definition or the terminology will be divided into many times according to the technology development based on the technology development. Currently, the cell production skill, especially the industry production, is very important to form the meat or form the food. Because only cell it's very small. It can't be called meat. Many cells can form the meat. But in the future, as the technology development such as Minghao or the other scholar experts, I think the technology diversity and the understanding of the people will becoming more and more colorful. Thank you.
Wonderful. Thank you so much. And Vineet, just any last words from you?
Yeah, first of all, it was very informative session for me personally. Dr. Minhao and Dr. Lee shared their research, and I was quite impressed by the work Mr. Minhao is doing and Mr. Lee is doing. So from the micro level to the level of 1,000 liters, like how much animal the cells they are producing. It was quite interesting for us. Like we are equipment manufacturer company, but to understand these details firsthand from them, it is quite interesting. And now coming to GFI and the future of cultivated meat, I have aligned my presentation primarily to the future because there is nothing to discuss more because the bioreactor which is available for NAB's production is being used for the cultivated meat also, and I don't think it is in synchronization with the requirement. Like as I told in the presentation also, we are working on a product which is very low cost, and we are using a very high capital intensive equipment to produce very low cost thing. So obviously there is a huge mismatch, and we need to develop a lot many things to be able to achieve our target to feed the 10 billion population in 2050. And to do that, a lot many inventions, a lot many innovations are required to be integrated into the bioreactor systems with the help of our biologists like Mr. Lee and Mr. Minhao, so that we can make such a bioreactor which can actually produce those cultivated meats at a price very competitive to the traditional feedstock. So this is my take on this.
Wonderful. And yes, thank you so much again for all of our speakers. Thank you to audience for attending. The YouTube channel will be up. I think so much great content was here, so we'll be able to share and continue to amplify your work. So I think beyond just those who attended today, I think many more people will be able to really learn from the material you shared. And I hope that this encourages further collaboration. Thank you again, everybody, for all the insights, and thank you to the audience. With that, I will wrap up our last session for the All Protein Dialogues for the year. So we'll see you in 2026. Thank you very much. Thank you.
Thank you.
Thank you.
Thank you.